Hello everybody, today we will be learning about genomics, transcriptomics, and proteomics. Genomics is a study of the entire genome and how it reacts with its environment. The entire genome is contained within the DNA sequence, which is contained within the chromosome of the cell. It contains all the possible information for any and every function that the organism might need. And not all of the genes will be expressed at one time. Some of them will never be translated within the life of that cell. Its contents are constant and do not change between the cells of the organism. Different genes are expressed within different cells and within the different life of the cell. So the code for everything is present in every single cell. Transcriptomics is a study of mRNA and transcription. This is the active part of the cell's genome. Transcriptomes are the method by which only certain parts of the genome are expressed in the form of proteomes. So basically, the transcriptomes are the mRNA molecules. They are the middleman between the genes and the proteins. So the genes from the cell's DNA will get transcribed into an mRNA molecule, and the gene coded by the mRNA molecule gets translated into a protein. Only the genes that are needed by the cell are transcribed into mRNA. So the transcriptome is only the active part of the DNA. It shows us which part of the DNA is necessary and which codes for proteins and which part is just filler, so the introns and insulators between different genes. The present mRNA molecules tell us what functions the cell needs to complete at that given moment. And this varies depending on which genes are being transcribed into the mRNA at a given point in time. So it shows us which parts are active, which parts are useful, and which parts the cell may never use depending on what type of cell it is. Proteomics is the study of the structure and functions of proteins in the cell. So it's essentially the study of the expression of the genomics, or the active parts of the DNA. The proteome is the implementation of certain parts of the genome, and they're the application of the transcribed genome. So they're the proteins formed by the mRNA molecules. And they vary constantly based on which of the transcribed genes are being translated into proteins, because only those coded for by the mRNA will be created, and the proteins present depend on which mRNA molecules have been transcribed from the DNA molecule. So which of the transcriptomes have been transcribed from the genome. So which proteins are in the cell at any given time depends on which mRNA molecules have been transcribed, and what has been transcribed is dependent upon what the cell needs to do. So the proteins tell us more specifically what the cell needs to accomplish at that given moment. To conclude, genomics is the entire set of information the cell will need throughout its lifetime, and a little bit extra. And the transcriptomics is only the useful parts of that entire set of instructions. We've skimmed through and weeded out the parts that don't really apply to us, and we've only kept the ones that do. It's only the helpful stuff. And then proteomics is the application of those useful parts of this entire set of instructions. So you've skimmed through everything, you've found the instructions that you need, and then you've made what you need to make from that set of instructions. Thank you guys for watching this video, and I hope you learned something new today.